Hello, everyone. I'm very pleased to present you our research on gene modifiers associated to colonic perforation in vascular ADS. I'm Xavier Jeunemaitre. I'm working in the Center for Rare Vascular Disease at Hôpital Pompidou in Paris, France, and working also at the Université de Paris at the Faculty of Health. I have no particular disclosure to report. So I won't present in detail the vascular Ehlers syndrome since you know it very well. It's a rare autosomal dominant disease with a defect in type 3 collagen with multiple minor signs. And so the real problem that I would like to present today are corresponding to the digestive complication and mainly colonic rupture, which can occur, as you will see, quite early in the life. Colonic perforation are the second major type of complication in VDS. You can see on the chart coming from the population that we are following in Paris that the vascular complication, of course, are more than 50% of the case, the first major complication, but the colonic perforation are occurring as a first complication in life of one individual with VDS in approximately 20 to 25% of the case. So it's a major, severe, and relatively frequent complication in this disease. It is so severe, actually, that usually it requires partial colectomy or even total colectomy. The mechanism of colonic perforation are not well known. They might correspond to a fragility of the colonic wall due to the lack of collagen. They might also correspond to ischemic parts of the colonic wall due to dissection or in the distal arteries or even thrombosis. Probably the two mechanisms are in play for that. Interestingly enough, as I said before, it's occurring early in life for patients with VDS. You see there the cumulative incidence that we observe in our cohort in France, and it can occur as early as 10 years of age, but usually between 15 and 50 years of age, you have all the complications occurring there. And it's also more frequent, a little bit more frequent in male than females. The second type of thing that's interfere with the colonic perforation is a type of mutation that people have. You probably know that you have two main types of variants occurring in the COL3A1, which are first the dominant negative variants corresponding to glycine missense variants or splice site variants, which actually are going to have a dramatic consequence on the folding of the collagen type 3 homotrimer. And the other type of variants, which are null variants, which have less consequences with approximately 50% reduction in the production of a normal collagen type 3. And these patients are usually more moderate in terms of the phenotype. And you can see there in red the two types of complications that are not occurring in patients with haploinsufficient variants, so null variants. This is the colonic perforation, which mainly do not occur in these patients, and also the pneumothorax as a pulmonary complication, which are not occurring in these patients. So it's important to see that it's only the dominant negative mutation that are going to have this kind of complication. Last thing about the genotype that we know is that even with the same kind of mutation, there might be a strong intrafamilial variability. You see, for example, on this pedigree that the first case which was diagnosed in this family was a young woman. She was approximately 20 years of age. And she had already, at the age of 18, arterial aneurysm or carotid dissection, which were diagnosed. And at 21, she had a colonic perforation, which needed partial colectomy. A few years later, her young brother had the same kind of complication with actually colonic perforation as the first complication of his VDS, which required totalectomy because of recurrence of this perforation. And surprisingly, you can observe that their father had very mild symptoms, even though he had the same mutation, and only arterial aneurysm. And so we do not understand very well what's going on there. And maybe that the genotypes or the genome of the mother might have an effect on this particular severity of phenotype in the children. So this is coming to the gene modifier hypothesis. If we are looking at the genetic susceptibility coming to the COL3A1 gene with the two type of variants, giving a phenotype with minor signs, arterial complication, colonic perforations, or pneumothorax, the idea is that you have on the left the possibility of different factors that can affect the phenotype: blood pressure for the arterial complication, lifestyles, environment, probably also mainly for the arterial complication, age and hormones, and we can discuss that later. One of these things that you can imagine that not only the COL3A1 gene, but also other genetic variants or even epigenetics can affect the phenotype. So this was the idea that we have pursued. What we have done is a genome-wide association study on more than 360 VDS patients coming to a large French cohort, which was established by the Radico Rare Disease cohorts, all in France. And from this patient, we had 300 patients with dominant negative variants and 69 patients with haploinsufficiency. So obviously, we are going to focus on these 300 for the digestive complication. We genotype them with DNA chip, which correspond to approximately 1 million of single nucleotide polymorphism, mainly frequent variants, but also some rare variants. And we applied sophisticated statistical models to perform this genotype analysis. 6 phenotypes were tested: acrogeria, age of the first severe complication, digestive complication, vascular complication with number of affected arteries, number of acute arterial complication. I'm not going to report on each of the phenotype. I'm going to report mainly on the digestive complication. The first thing that we observe in this course is that some of the phenotypes are going together. For example, on the left upper part, you can observe that there is a correlation between the presence of digestive complication, for example, and pneumothorax. On the other side, you can see that you have correlation between the arterial complications themselves, but there is no correlation between the presence of arterial complication and presence of digestive complication. And the minor signs are going together. So this was useful for us also to try to establish a model to analyze the genotype-phenotype correlation. And I'll present there our main results for the colonic perforation. What we observed from 54 patients which were positive for this phenotype compared to almost more than 250 negative for this phenotype is that there was a genome-wide significant association on this Manhattan plot with chromosome 4 locus, with even an odd ratio of 7.5, so a strong odd ratio with this corresponding locus to have a complication compared to those who had not this polymorphism, at least at the same frequency. And we have also some suggestive loci on chromosome 2, 3, 6, and 12 that you can observe there, and which obviously are suggestive but not confirmed yet. It's important to know that for the moment, these loci do not correspond to particular genes. what we have for the moment are single nucleotide polymorphism, which are in intergenic regions. So they are not focusing on one particular gene, but probably in regions which are regulating the expression of one given gene or several genes. And obviously there is a long way to go to understand the association and the causality of this association. So we need replication for that. We need other cohorts, and to be sure that the association stands up with this chromosome 4 region, but also with the other loci. And we can genotype these new patients by directly looking at the loci or genotyping all the genome to test not only the digestive complication, but others. And obviously the goal is to make a meta-analysis of these different studies. And you have there the emails that we have and the contact to try to establish as soon as possible and as largely as possible this association at an international level. So our main take-home message is first, the colonic perforation are the second major severe complication in VDS patients. They occur in patients with dominant negative variants, missense glycine or splice site mutation. They occur at early age, mainly between 15 and 40 years of age. There is no association with other signs of complication except pneumothorax. There is no particular environmental trigger which is identified yet to explain this colonic perforation. And gene modifiers may play a role, and the first GWAS analysis that I just presented there has identified suggestive loci. And we think that this is the first step towards an even more personalized medicine for VDS patients. We'll end up with acknowledging the people working with me on that topic. Anne Legrand, who is doing her PhD thesis on that topic. Barthélemy Caron has done all the statistics, Michael Frank for the phenotype, and all the people and also the French radical registry and all the centers in France which are devoted to this disease. Thank you very much for your attention.